Michael. So in this lecture, we'll be talking about something that is really exciting. We'll talk about the human genome, components of the human genome, the complexity of the human genome. What's really fascinating is that it changes, the information changes every single day. What's frustrating is that every single year I have to change my slides, but yet it's really exciting. So you can go back to the Cooper's textbook to certain pages, but pay attention to the information in the slides themselves. Okay, so I'll start with some little history so that you can keep up with what you hear in the news. So inshallah, you hear the news. So in 1990, there was this international project that was launched, and they said that it would take about 13 years, probably 15 years, to sequence the human genome, and it costs about $3 billion, a lot of money. And by the way, you can do the same thing nowadays for like 500 JDs or $500. So just imagine the advancement in technology. So in 1990, it would cost $3 billion to sequence the human genome. Now it costs about $500. So anyhow, so it's an international project. 7 countries participated in it, and the objective was to sequence the human genome. So what do we mean by sequence? Sequence DNA, if I say DNA sequencing, it means knowing the order of nucleotides in a piece of DNA. So what we wanted to know was basically the order of nucleotides in the human genome. So it's like taking a book, and if it says, for example, THE, so knowing the order of letters, so THEBOOKIS, whatever, okay? So it's basically the book is. So basically what they wanted to know is the order of nucleotides, the sequence of nucleotides, hoping that they would be able to read this DNA, trying to decipher useful information out of the letters, okay? Turning letters into words, in other words. So anyhow, so and to map the identified genes, that is to know the location of genes. For example, on chromosome 1, on this position, this location, there's this certain gene. On chromosome 2, there's this gene right here, there's this gene next to it, and there's another gene that is far away from it, and so on. So it started in 1990. A draft was published in 2001. They made a big deal out of it. So you can see it here in these 2 journals. Okay, so these are 2 of the most prestigious journals in the world in terms of science, all of science, Nature and Science. In 2004, Bill Clinton, the US president, announced completing the sequencing, the Human Genome Project. Okay, and these 2 pioneers right here, Greg Venter and Francis Collins, they were involved in this process. Really long story, fierce competition between the 2, but regardless. So. But it was a lie, actually. You know, they announced that they completed the sequencing the human genome, but it was a lie. They only finished 92% of it, because the rest of the human genome was made of repetitive letters, okay, and sequences, and that gave them a huge problem. So what was the purpose of knowing the sequence of the human genome? Basically, they wanted to determine the number of human genes. So they thought at first that the human genome, and genome, by the way, means the total collection of DNA in a cell. Okay, so if I say the genome of a cell, the total component of DNA. If I say the human genome, I mean, you know, the DNA that we share, okay, and so on. So they wanted to determine the number of and location of genes, like I said. Now, at the same time, in launching the project allowed for advancement of science in terms of technology. And that was huge, by the way. So every single day they would invent a new instrument or new technique, or in terms of computer science, in terms of developing programs, computer programs, to analyze these words, to turn them into meaningful letters. That was huge, by the way, and that's why, I strongly believe that the future lies in IT. The future lies in analyzing big data, okay, turning something that is meaningless into something that is meaningful, turning data into information. So, and it worked, okay. So like I said, 15 years, 20 years ago, 25 years ago, it would cost about $3 billion to do the same thing that would cost these days only $500. That was huge, by the way. But the clutch, or the important point is really turning the data into information. So along the way, with the invention of new technologies and new knowledge in terms of the human genome, they were able to also sequence the genomes of other organisms, other species as well. So right now they would just take, and actually Greg Venter with his company, they would go into the sea, they would just take seawater randomly from different layers of water, and they would extract DNA, take DNA from all of these cells and organisms and whatever, randomly. I'm not talking about fish, I'm just talking about small organisms, okay? And they would sequence the whole thing, the whole DNA, okay? They're trying to learn the DNA sequences of other organisms, unknown organisms, hoping that they can use them in industry to turn, I don't know, like dust into gold or something. So they were able to do that as well, okay? And at the same time, they would compare other organisms' DNA to human DNA. So, but that also opened the door, knowing the sequence of DNA would open the door for legal and ethical issues. So let's say that I sequence your DNA and I find that there's a mutation, there's something that causes, okay, let's not talk about it here. Let's say that I sequence someone's DNA and I discovered that there is a mutation, there's something wrong in the DNA that would cause this person to develop a certain disease after a period of time. Should I tell that person or not? Is that ethical? Should I hire this person or not, knowing that this person might develop a certain disease? So there are a lot of ethical and legal issues associated with the information that we get out. Oh, something else. Should I marry your daughter or not? Well, not me, of course, but should I marry your daughter or not? So, think about all of these things. Okay, so this is an old slide, okay? But just to show you a sort of like comparison between human DNA with with the DNA of other organisms. So if you look at the number of nucleotides, and this is not for you to memorize, okay? So do not memorize. So if you look at human DNA, we have about 3.2 billion base pairs, or 3.3 or so. If you look at the mouse genome, it's about 2.6. Well, if you look at the number of genes, they're sort of like similar to each other, by the way. Okay, so that's interesting. If you look at the number of chromosomes, there is some similarity, but there are major differences as well. So you look at the fruit fly, it's one of those organisms that are well studied. There are about 13,000 genes. Well, look at the roundworm, it's about 19,000 genes. Okay. By the way, the human genome, we have about 20,000 genes right now. I'll give you the most recent number in a few seconds. So it's really similar to the number of genes in the roundworm. So, so what do we do with this information? Is this information important? Really important. So This just shows the number of base pairs in different genomes, mammals, birds, reptiles, bacteria, archaea. Archaea is like the third branch of living organisms. So you have eukaryotes, prokaryotes, and you have archaea. Archaea is a combination of prokaryotes and eukaryotes. Well, if you look at plants, so that shows different plants like rice and lily and wheat. So if you look at the number of base pairs in different genomes, so you can see the variation right here. So in humans, we have 3 billion base pairs. Mammals, it's about 1 billion to about close to 10 billion base pairs. Again, do not memorize. If you look at the variation of base pairs or genomes, genome sizes in protozoans, you can see huge variation right here. Amoeba, really simple organism, much simpler than human, but notice that the size of the amoeba genome is much larger than the human genome. So it gives you some information, but not a whole lot, because again, there's a lot of variation. Just this information is for you to just have fun with it. Well, there's an interesting concept, and that is DNA homology. So why is it important that we learn DNA of other organisms? Because we can use them, we can learn from them. There's huge similarity in DNA homology. There's high DNA homology. What do I mean by homology? Sequence similarity. So if you compare the sequence similarity of the human genome with the mouse genome, for example, with the chimp genome, there's huge, or let's say significant homology or similarity in the sequence, and that gives us really important information. Not only in terms of evolution and stuff like that, but in terms of the function of these DNA sequences, the function of genes. So if you look at, for example, a certain sequence, this is in human versus rabbit, rat, and mouse. So you can see that the colored regions is where you have huge similarity, and these are the functional regions in the DNA. Okay, so you can see that whenever you see this light right here, this line, it means that there's similarity in the sequence. So let's zoom in, for example. Where is it? There's no zoom when I record lectures. But whenever you see a line right here, it means that they're similar. So these sequences, see the letters, the order of letters is really similar among the different organisms, and that tells you that this sequence is important. Okay, so if I discover a gene in mice, I would expect that the same gene to exist in humans. So what does make humans humans first if we share the same genes? Well, it's more complex than what we think. And we'll talk about it throughout the lectures. So anyhow, so if you look at homology again among different organisms, you see that there's a high similarity. But the differences are probably what make us different. So this is a nice flow chart that shows the different components of the human genome. I have updated this flow chart based on recent information. So let's look at it like this. So here we have the human genome, the total DNA content in a cell. And that includes actually two sources. First, we have the nuclear genome, the DNA in the nucleus itself. And we have the mitochondrial genome as well. So we do have mitochondria, of course. They generate energy in our cells, and they contain their own DNA. And this DNA contains some important genes for the respiratory chain reaction that takes place in the mitochondria, and that is responsible for generating energy. So it's really a small genome. It's about only 16.5 kilo base pair. That is 16.5 thousand base pairs. So, but if you look at the human genome, it consists of about 3.3 billion base pairs. Okay, so remember, base pair means that we have two strands. Each one of them contains 3.3, and the other one contains also 3.3. Okay, so we're talking about base pair right here. So if you look at the nuclear genome, it consists of genes and gene-related sequences, and it contains extragenic or intergenic. Okay, so we'll talk about genes and gene-related sequences first, and they constitute about 30% of the human genome. Now, the protein coding sequences, the sequences that are responsible for making proteins, it's only 2%. Some sources actually say 1.5%. So that's really little, okay? I remember when I studied, when I was your age and I was studying in the US, they kept saying the rest is junk DNA. 98% of the human genome is junk DNA, and I hated this term. It just didn't make sense that it was junk, and actually it turns out to be really important, and we'll talk about it in a second. So we have genes and gene-related sequences. Genes constitute about 2% of the human genome. About 28% of the human genome is non-coding. What do we mean by non-coding? It means that it does not code for proteins, or it does not get transcribed. It doesn't get it doesn't code for RNA. It doesn't produce RNA. Okay, so they call it non-coding sequences. These sequences mainly contain introns. We'll talk about introns later on. So when you have transcription of messenger RNA, you have exons and introns. Introns are removed, exons are connected to each other, and then they get translated. Okay, so introns are the portions, the parts of the messenger RNA that are removed, and they get discarded, but they do not get discarded actually. They are still important. So 28% of the human genome is basically introns. We have pseudogenes and other sequences as well, like untranslated regions that are part of the messenger RNA, but they do not get translated into protein, and we'll talk about this in the translation lecture. Okay, so let's talk about extragenic or intergenic sequences, which constitute about 70%. By the way, these numbers are approximate, okay? So you can memorize them if you want, but they will keep on changing year after year. So extragenic, like outside of genes, okay? Intergenic means like between genes, okay? So they constitute about 70% of the human genome. That's huge, right? Now, about 5% is basically unique or low copy number. That is, well, they are unique sequences, okay? They're different from each other. So that's little. About 55% is basically repetitive DNA sequences. The same sequence, but they are repetitive. They are present in different places, or they are repeated. About 10% is other. Okay, I couldn't find a good definition for other, or it's complex. I don't want to get involved in these other sequences. So let's talk about the 55% right here. So these are moderate to highly repetitive sequences. Now these sequences can be tandemly repeated, or they can be dispersed. What do we mean by tandem? Tandem means that it's the same sequence repeated one after the other. So it's like saying ABC, 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 ABC. Okay, so it's within a DNA fragment, and they are repeated one after the other. Now dispersed means that they are the same sequences like ABC, but they are located on different places, on different chromosomes. So they are dispersed. So they can be placed. So they are located on the same chromosome but on different places, or they are placed on different chromosomes. Okay, so the ABC can be found like on chromosome 1, chromosome 10, chromosome 21, and so on. So they are dispersed. Okay, so if you look at these sequences, they constitute about 55% of the human genome. That's huge, more than half of the human genome. They are, they can either be tandemly repeated or interspersed. Well, let's talk about these tandem repeats right here. That is the same sequence but repeated one after the other within the same place or small region. So that these regions, these sequences constitute about 10% of the human genome. So memorize these numbers, by the way, relatively, okay? So know these numbers. Many of them are satellite. They're called satellite sequences. Satellite means that is, they are like on the side, okay? Because these sequences, when they were separated from other DNA sequences, they were like located or separately or in different position. I'm not going to get into details, but these are, many of them are satellite DNAs, DNA sequences, and they are located in centromeres and telomeres. So we'll talk about centromeres and telomeres in a few seconds. Some of them are known as mini satellites. So mini means small, mini satellites. Also known as VNTRs. We'll talk about these as well. And others are known as microsatellites, even smaller than mini satellites. Okay, so we have microsatellites or short tandem repeats, STRs. Okay, so let's talk about the interspersed DNA, repetitive DNA sequences. There are two types. We have DNA transposons. Ooh, what do we mean by transposons? Means that these are movable DNA sequences. That is, they change locations. So 3% of these transposons originate from DNA. And we have retrotransposons. About 42% of the human genome is composed of retrotransposons. Again, transposons, movable elements, movable DNA sequences. That is, they change location. So what does retro mean? Retro means that they originated from RNA viruses. Okay, so about 42% of our genome originated from RNA viruses. This RNA got converted into DNA, and the DNA got integrated into our DNA, into our genome. So you can imagine that 42% of our genome really originated from RNA viruses. Amazing. And there are different types. We have what is known as signs, 13%, lines, 21%, S stands for short, L stands for long. We'll talk about them at the end of this lecture. And we have retrovirus-like elements. They look like retrovirus genetic material, but not exactly, okay? And that constitutes about 8%, okay? So this is how our genome looks like. Now, what's really interesting is that 5% of the genome contains sequences of non-coding DNA, DNA that is not transcribed, but they are highly conserved, okay? Highly conserved, that is meaning that these sequences are similar to sequences found in mice and chimp and cows and cats and dogs and lions and you name it. And that tells us something, that these sequences, even though they're non-coding, even though they're not transcribed into RNA, they are conserved because they are important. Okay, so, and that's why they have been conserved, kept throughout evolution. When scientists were near completing the Human Genome Project, they started another project. They said, okay, so we're gonna finish the human genome. So what can we do with this? So they initiated another project and they called it the Encode project, which means Encyclopedia of DNA Elements. And by the way, whenever you hear the word element, it means DNA, okay? Whenever you hear the word factor, it means protein. So we're talking about protein factors, and we talk about DNA elements. Okay, so so they started this project trying to turn data into information, and they were able to generate some really interesting findings. There is one, the number of protein coding genes is only about 20,000. Well, actually, it's going to be less than that. I'll show you in a second. But just imagine that. I do remember when the Human Genome Project started in 1990, because that's when I first started studying in the US. They thought that the number of genes in the human genome is about 100,000 genes, compared to what they knew of bacteria or yeast or smaller organisms or simpler organisms. So they thought, well, human DNA must be, or human genes must be much higher than bacterial genes, than the number of bacterial genes. So they thought that it would be about 100,000. And along the way, as they sequenced the human genome, they said, it doesn't look like 100,000. It must be 75,000 genes. Then they said, it looks like 50,000 genes. Then it was like 40,000 genes. Then the last number was like close to 20,000 genes. And that was mind-boggling. That was really shocking to scientists. We only have 20,000 genes, just like fruit flies and yeast and mice and chimps. Well, it looks like it. Well, what's really important about the Encode project is that they were able to find out that 75% of the human genome is transcribed into RNA. They thought that it was only 2% of the human genome that is transcribed. But it turns out that about 75,000 of the human genome is transcribed. We still cannot figure out what the 2% does. Okay, so now we have to deal with 75%. This is huge, by the way. That's huge information that we have generated in the past 10 years, and we don't know what to do with this information, by the way. And we'll talk about some of these RNA molecules. So, and about 8% of 8% of the human genome generates transcription factors. Okay, we'll talk about transcription factors in the transcription lectures. That is, what they do is that they regulate transcription. Okay, so these are the information that we have found out so far. So about 80% of the entire human genome is relevant. It's not junk like what we thought. So how about the other 28%? Well, we don't know yet what. Well, let's deal with the 80% first. Okay, so this is huge, by the way. Okay, so let's look into this slide. This is important. And there was this study, the complete sequence of a human genome, that was published on March 31st, 2022, really recent. And it made huge splash in the news, in science, all over. Really big. Now, they didn't really sequence the whole human genome. They didn't finish, they didn't complete the sequencing the human genome, because they still have this miserable Y chromosome that has not yet been sequenced completely. Now it has been sequenced, but we still have portions of the Y chromosome that have not been sequenced. Why? Because these are sequences that contain repetitive DNA sequences. So these scientists were able to sequence everything, all DNA, including repetitive DNA sequences, except for the repetitive sequences in the Y chromosome. And they found out the following. The number of genes in the human genome is about 63,000, close to 63,000 genes. Now, only 19,969 of these genes are protein coding. They produce protein. They produce messenger RNA, in other words. Okay? Now the number of transcripts, what do we mean by transcripts? That is RNA molecules. This is what transcripts mean. The number of RNA molecules that are produced from the 63,000 genes is 233,000 RNA molecules, different RNA molecules. About 86% of those are protein coding. Okay, that is, in other words, messenger RNA. So what's the difference between this number right here and this number right here? Well, right here we have about 19,000 genes that produce RNA molecules, or premature or messenger RNA molecules. They get processed differently into 86,000 mature messenger RNA molecules. We'll talk about these later on. Okay, so we have these processed differently into 86,000 different messenger RNA molecules that produce different proteins. Okay, so we have 63,000 genes, but only 19,000 of them produce protein. What about the rest? Well, these are called, they produce non-coding RNA. They produce non-coding RNA. That is RNA that does not get translated into proteins. They have other functions. And these include some of the RNA molecules that I talked about before in the first lecture, like microRNA, long non-coding RNA, circular RNA. We'll talk about some of these later on throughout the lectures. So in the following slides, what we'll do is we'll talk about some of the components of our DNA, the human genome. And we'll start talking about the tandem repeats. So first, let's talk about satellite DNA. That is DNA regions that can be, when fragmented, they can be separated from the rest of the DNA fragments or DNA regions of the human genome. Now, these satellite DNA molecules, they represent regions that are repeated, that have a size of 5 base pairs to 300 base pairs, and they are repeated millions of times. And they are mainly localized in centromeres and telomeres. So centromeres are the regions that sort of like they exist in the middle of chromosomes, but you'll learn in genetics that they can be located in different regions in the chromosomes, and they separate chromosomes into 2 regions, okay, like the large arm and the short arm. Anyhow, so they exist right here, and these centromeres are really important for cell division. Okay, when you have these fibers connected to chromosomes, they are connected to centromeres. Now, these centromeres contain a lot of repetitive DNA sequences. Now, telomeres exist at the ends of chromosomes, at both ends of chromosomes. So telo means end, okay? So and they also they are composed of DNA sequences that are highly repeated. Now centromeres are AT-rich repeats. They are composed of about 171 base pairs. They're called alpha satellites, and they are unique to each chromosome. So each chromosome has its own sequence of centromeres, and we can take advantage of probes in labeling centromeres and telomeres. So, for example, centromeres of different chromosomes have different sequences, so we can use the probes to label centromeres of different chromosomes. We can do the same with telomeres, but telomeres of different chromosomes have the same sequence, so we can use the same probe to label the ends of chromosomes. So you can see here, for example, we can use probes to label centromeres. Okay, so and we do the same with telomeres. So we use probes to label the ends of chromosomes, as you can see here. So this is a technique known as fluorescence in situ hybridization. So what we do is that we take a DNA probe. We already discussed what a probe means. We label the probe, and the probe is homologous. That is, it has a similar sequence to the probe that to the DNA that we are targeting. Okay, so remember DNA homology. It's homologous, so it binds to centromeres, for example, or it binds to a certain on a certain location on the chromosome. So here we can even label certain genes to see where they are located on chromosomes exactly, or we can probe for centromeres or telomeres, like I said in here. So this technique is known as fluorescence in situ hybridization. What does in situ means? It means in place, okay, in an exact location or in an exact place. So it's not, so again, that's fluorescence in situ hybridization. Now, there are other repeats, and we talked about these repeats before. These are the repeats that exist in telomeres. Now, telomeres are, again, they exist at the ends of chromosomes. Telo means end. Okay, so these are the regions that are located at the tips of chromosomes. As this is another image of fluorescence in situ hybridization, where you have telomeres being stained. Now, of course, telomeres, as you know, they're composed of a repeat, that is TTA-GGT, and or TTA-GGG without the T at the end. It doesn't matter because it's a repeat. Anyhow, and these are repeated like hundreds to thousands of times at the termini of chromosomes. And there is a 3' overhang. Okay, so right here at the end of the chromosome, that's where you have the lagging region being extended, right? So you have this extension right here that is created by the telomerase. Okay, now remember how I gave you the example of telomeres after being elongated, and in order to make sure that chromosomes are stable, it's like sewing a button in a shirt or something. So at the end, you have this extra thread, and you just tie it, okay, so that the button would stay in its place. So this is the same thing. You can visualize it as the same thing. So at the end of the chromosome, it's tied up, and this is done also with the help of a protein known as shelterin. So what it does is like it shelters chromosomes. Okay, so it protects the termini of chromosomes from degradation. Now there's something fascinating that I found, and it's not in your Cooper's textbook, and that is telomeres can be transcribed. Now, in the past 15 years or so, I've been teaching molecular biology, and I always said that telomeres and centromeres are not transcribed. They are heterochromatins. They do not contain any genes, but actually now we know, because of the Encode project and other work, and development of technologies, that telomeres can be transcribed, as well as centromeres, but I didn't talk about centromeres being transcribed in the previous slide. But telomeres can be transcribed. Okay, it's amazing. So you have the formation of a telomeric repeat containing RNA, also known as tera. And this RNA it belongs to the class of long non-coding RNA molecules. And these RNA molecules that are transcribed from telomeres, they have important functions in maintaining the integrity of chromosome termini. So they work similar to sheltering. They can also regulate telomerase activity. They maintain the heterochromatic state of telomeres. So remember, we have euchromatin and we have heterochromatin. Heterochromatin is where you have DNA highly packed. Telomeres are maintained in the heterochromatic state, very highly packed, and this RNA tera also can protect DNA from deterioration and degradation or fusion with neighboring chromosomes. Now, there are genetic abnormalities whereby you can have chromosomes fused with each other, and it would be fascinating to know what the role of tera is in chromosomal fusions. So this is really fascinating and up-to-date information. Now, mini and microsatellite DNA sequences, they are highly repeated, okay, but they differ in size. So, for example, mini satellites are also known as variable number tandem repeats, VNTRs. So you have a sequence that is made of, let's say, about 20 base pair sequences, 20 to 100 base pair, and this sequence is repeated several times, and they are tandemly repeated, so they are close to each other, one after the other. Microsatellites are repeats that consist of about 2 to 10 base pairs, and they can also be repeated several times, as you can see in this example right here. So mini satellites, or VNTRs, they are each repeat contains about 20 to 100 base pair sequence, and it can be repeated 20 to 50 times. On the other hand, microsatellites, they are repeated 10 to 100 times, and they have this size, 2 to 10 base pairs. Okay, so you can have, for example, since we are diploid, or our cells are diploid, you can have on a chromosome this repeat right here, the CA repeat, so CA, 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 and it's repeated 16 times. On the other chromosome, you can have the same exact repeat, but it's repeated 14 times instead of 16 times. So you can have a lot of variation, even individual. Now, this is a cause of polymorphism, that is, different shapes of DNA sequences. So you can have, for example, different sequences of VNTRs and STRs on the on homologous chromosomes, like I showed you in the previous slide. And this has been helpful actually in forensic testing, because we can differentiate samples of individuals from each other based on this polymorphism. So here's an example right here, a few examples. So you have individual A, you look at STRs or a certain VNTRs. On one chromosome, you have 3 repeats. On the other chromosome, you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 repeats. Okay. Now, on the other hand, you have individual B on homologous chromosomes. The same exact sequence exists on the same location, on the same chromosome exactly, except that the number of repeats is different. So on both chromosomes, you have 6 repeats rather than 3 or 10. So this is individual A. You have this repeat is the one that is repeated 10 times, and this is the one that is repeated 3 times. Now, in this individual, there's only one band. If we do RFLP, or if we do PCR, and we'll talk about PCR later on, and there's only one band, which is the 6-time repeated VNTR. So look at these different individuals looking at a certain VNTR or STR. You can see how each individual has his or her own molecular pattern, and this is what is known as molecular profiling or fingerprinting. Now we can use VNTRs and STRs in paternity testing, in forensic medicine, in diagnostic medicine as well. And this is an example. Okay. So right here in this family, okay, this is known as a pedigree. So what you have in here, you have square representing a male, circle representing a female. So you have male and female, they're married, they are connected to each other, and they have this little daughter. Now, and you have another family, female, male, so they get married and they have this boy, and this boy gets married to this girl right here, and they have lots of kids, or أولاد, in other words. So they have this female, male, 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 female, male, and female. Okay. So if we look at this family, and if we look at a certain VNTR, for example, we would notice that there are 4 different alleles. So alleles is basically type of a gene or type of a DNA sequence. So they have 4 alleles. Allele number 1 is when they have 6 repeats. Allele number 2 is having 4 repeats. Allele number 3 having 3 repeats, and allele 4 is having 1 repeat only, or one of such sequence. So by doing RFLP or by doing PCR, we get the following. So these patterns represent the person that is located on top of them. Okay, so here we have the grandfather, okay, of the first family, and this grandfather has allele number 1 and allele number 2. So he has these 2. Now this grandmother has allele number 2 and allele number 3. So you would expect this daughter to have one of these 2 from her father and one of these 2 from her mother. And that's true. In fact, she has allele number 1 from her father and allele number 3 from her mother. Look at this family now. Let's look at this grandmother right here. She has alleles 2 and 3. Look at this grandfather. He has alleles 1 and 4. So you would expect this boy to have either one of these 2 alleles and either one of these 2 alleles. And that's true. He got allele number 2 from his mother, and he got allele number 4 from his father. Okay, now let's look at these individuals right here, these children right here. So you would expect them to have either alleles 1 and 3 from the mother, or alleles 2 and 4 from the father. And you can see the pattern in here. Okay, so this is from the mother, this is from the father, and so on. Now, so how can we differentiate these 3 individuals? How can we differentiate these 2 individuals in this case? Well, what we do is we look at another allele, or we look at another VNTR. So or we look at another STR, and so on. So in forensic medicine and paternity testing, they do not rely on one single VNTR or one single STR. Rather, they look at multiple VNTRs and STRs. Okay. Now, so we can use this for paternity testing. Same idea as like I showed you before in the previous lecture, or in this one right here. You can see the pattern between the parents versus children. Okay. So now, one major feature of the human genome is variation. And variation is not only in VNTRs and STRs, rather variation can also exist in single nucleotides, and this is known as single nucleotide polymorphism. So this is a major source of polymorphism. So basically, the idea is that at a single location on a chromosome or DNA, you have the same exact sequence, except that there is a difference in one nucleotide, and this is single nucleotide polymorphism. Okay, so when is a change considered a polymorphism versus a mutation? Well, it is known, this is the thing, if it exists in more than 1% of the general population, if a variation, a genetic variation, a SNP as we call it, exists in more than 1% of the general population, we call it a polymorphism. If it exists in less than 1% of the population, then we consider it a mutation. Okay? So this is an important point that you need to know. Now, so these SNPs are really spread throughout the human genome, and there is one SNP for every 300 base pairs. So we have a total of about 10 million SNPs in our genome. Okay, 10 million SNPs, that's a lot. But only 500,000 are important or relevant. So what do we mean by important or relevant? Well, let's see here. So here we have examples of SNPs. So you have an individual, for example, having this sequence. This person has, at a certain location that is known to be variable among individuals in a population, this person has G and G on both paternal and maternal alleles. So we said that this person is homozygous for this particular SNP. Okay? Now let's look at another sequence, another location, and we would notice that the paternal allele contains an A, the maternal contains a C. Okay? So we say that this person is heterozygous for this particular SNP. Notice that the distribution of this SNP among individuals in a population is that A exists in 90% of the population versus C, which exists in 10% of the population. So we call this a minor allele. Now, these SNPs in individuals in a population, the G exists in 51% of a population versus T, which exists in 49% of the population. But this individual does not have a T, obviously. But notice that all of the other sequences are exactly the same except at these two positions. Okay, so this is individual 1 looking at a SNP in chromosome 2. Different individuals, 6 individuals. This person has T and C. This individual, same location, same SNP, same position, has C and C. This person has T and T. This person has T and C. So, and this person has C and T, and this person has C and T. Okay, so you can see that these SNPs, mainly they are either T or C. Okay. How are SNPs important? So I said that 500,000 SNPs are relevant. What do we mean by relevant? Well, these SNPs can be divided into two types. They can either be linked or they can be causative. What we mean by causative is that they cause a phenotype. Okay, so they cause a certain phenotype. For example, maybe a phenotype in I don't know, skin color, maybe a phenotype in something that you can see. Maybe sometimes it's a phenotype that exists when a certain condition happens. So for example, you have two individuals, they look maybe brothers, they look exactly the same, except that when one of them takes a medication for headache, like paracetamol, for example, you see one of them needs just one pill, the other one would need 2 pills. Okay, we wouldn't be able to know unless they go through this certain circumstance or certain condition. So this is a variation. Now, this variation can be relevant, well, because of their need of different doses of medications. So that's what we mean by it causes a phenotype. So that's a causative SNP. Now, it's not a major thing. It's not a mutation that causes a disease. Rather, it causes a certain phenotype. So causative SNPs can be located in non-coding regions, and we call them non-coding SNPs. So they can be located, for example, in the promoter region. They can be located in a regulatory sequence, meaning that a sequence of DNA that regulates, for example, gene expression, protein production. Or SNPs can also be in the coding region. So they exist in the DNA sequence that results in that influences protein production, or the sequence of amino acids in a protein. So we call it a coding step. This may change the amino acid sequence. Sometimes this change may not be that important. Okay, so it's not like the protein is defective. The protein, or let's say an enzyme, it might be about 50% less efficient than if it has the other SNP. Okay, and we would not be able to know the person is totally normal. Now, or SNPs can be linked, meaning that they do not cause a phenotype, rather that they are linked to a certain phenotype. So we notice, for example, if we look at individuals having a certain disease, let's say, we would notice that a lot of individuals have, with this disease, have T's. Okay, it's either C or a T. And these individuals that are affected with a certain disease or condition, they have a T. Okay, whereas in healthy individuals, very few of them have a T, okay, but most of them have a C. What does that mean? Does that mean that if we change a C, this C to a T, let's say, or if C is mutated to a T, these individuals would be affected? No, not necessarily. Okay, does that mean that if we change T to a C in these individuals that are affected with a disease, does that mean that they would not have the disease? Again, no, not necessarily true. But what it means is that there is a high frequency that individuals having a disease would have a T. It's just a probability. Okay, so if you know that someone has a T, you would say, you know what, there is a probability that you might develop a certain disease. It does not mean that for sure he would develop that disease. So what does it mean that it's linked? It's linked to a phenotype, yes, or it's linked to a gene. Maybe it's located near a gene that causes that disease. So when the gene is inherited, the SNP is also inherited along with it. Okay, so that's what linked means. And I hope it's clear. Well, how about interspersed repeats? These are the repeats that make up the majority of the human genome. Now, these interspersed segments are also known as transposons, which means jumping genes. Okay, so these are really segments of DNA that can move from one place to another in our genome. And it, by the way, it does not mean that as I'm talking to you right now, I have my transposons jumping or changing places. Same thing with you, by the way. So it's just that these are regions that can change place. Now, the majority of our transposons do not move. Okay, they have lost the ability to move and change places, and they do need help somehow. This is still under extensive research. But in other species, by the way, transposons can jump and can change places like in pigs, for example, in mice, in plants, and so on, but not in humans. It's not that common. Now, there are two types of transposons. We have what is known as DNA transposons, and they make up about 3% of our genome. Okay, but the majority is basically RNA transposons or retrotransposons, and they originated from retroviruses, viruses with RNA being the genetic material. So RNA transposons, retrotransposons, they make about 42% of the human genome. The majority is known as long interspersed elements or lines, and they make up about 21% of our genome. Then we have short interspersed elements or signs, and they make up about 13% of our genome. A great example of signs is something known as an ALU sequence, and it's known as ALU because it can be cut by a restriction endonuclease known as ALU. And the size of ALU is about 300 base pairs, and it's the most common sign in our genome. And actually, I was working, when I was a student, I was working on a gene, and we were puzzled by having an extra RNA piece that has a size of 300 base pair, and we don't know exactly what it was, and it turned out to be actually an ALU transposon that was part of the gene, and that was astonishing, and we published a paper on it. Now, we also have another example of retrotransposons is something known as retrovirus-like elements, and they make up about 8% of the human genome. Now, so these transposons have lost in the human genome, they have lost the ability to move, to change places, but we can have some active transposable elements that can sometimes move, and when they move they can cause diseases, and there are diseases that are related to retrotransposons changing places. Examples include hemophilia A and B. Hemophilia is basically a blood disease that results in excessive bleeding, so blood does not coagulate normally, so the person keeps on bleeding. You have severe combined immunodeficiency. There is porphyria, which is related to heme metabolism. Cancers can also be caused by transposons moving. We have Duchenne muscular dystrophy, another example. So why do they cause diseases? The reason is that when you have a transposable element, when you have a transposon changing places, it can insert itself within a gene. So what it does is that it disrupts the gene. So the gene does not function anymore. Okay, and if it produces a protein, then this protein is not functional. Or it can be, it can insert itself close to a gene, so the gene is not regulated appropriately, meaning that the gene can be overexpressed, so you're producing a lot of RNA and protein, or it can be underexpressed. Okay, and so there's failure to express the gene or to encode the protein. Now, otherwise, and that would cause a certain disease depending on the gene that is disrupted. Now, otherwise, the transposable element, this transposon, can insert itself far away from an important gene, and in this case, there would be no effect. But now, knowing that 75% of the human genome can be transcribed, and up to 80% of the human genome is functional, this can actually change the equation. Okay, so but that's what it is.